So any questions at all, just let me know because it's going to cover a lot of material. And we're going to start with the characteristics of cells and life. What makes something alive? So all living things, whether they're single cells or multicellular, are made up of cells. They share some common characteristics. They have a shape. The shape can be like humanoid shape. The shape can be spherical, cuboidal, cylindrical, these are bacteria. They have internal content, something has something to be inside the cells. Okay, that's cytoplasm, surrounded by a membrane that holds it together. Cytoplasm suspends all the different organelles and things inside the cell, nucleus and all your functioning equipment. DNA chromosomes, RNA, or DNA chromosomes have some kind of genetic information to transfer from one generation to the next. Ribosomes, metabolic capabilities. Ribosomes produce proteins, enzymes for the body. Keep things going. Metabolic capability, you have to be able to metabolize food. You have to be able to make energy. There's two types of cells, eukaryotic and prokaryotic. We talked about those before. Prokaryotic means before the nucleus. Okay, so bacteria are prokaryotes. Eukaryotics are human cells. They have a nucleus. Okay. So eukaryotic cells are animal cells, plant cells, fungi, and protists. Okay. They contain membrane-bound organelles that compartmentalize the cytoplasm and perform specific functions. There's different things in the cytoplasm, different organ. Some are membrane-bound, some are not, like mitochondria, Golgi apparatus, things that help process your proteins that are performed. Okay, there's a double membrane nucleus with DNA chromosomes in it. Okay, it's a double membrane and there's pores in it so that the DNA can copy itself into RNA and it can leave the nucleus in the cytoplasm where it binds to ribosomes and makes proteins. Prokaryotic cells are no nucleus. Okay, those are your bacteria and archaea. Okay. So there's no nucleus or other membrane-bound organelles inside the prokaryotes, bacteria cells. So this is some of the characters that cells have to have. Reproduction. They got to be able to reproduce. Any organism has to be able to reproduce. Has to metabolism and enzymes. So things can happen in the body at normal temperatures. Okay. You have to have DNA, hereditary. You know, transfer qualities or characteristics to next generation. You have to be able to grow and develop. You have to be responsive. Okay? You have to be able to respond to the environment. If something is bad, you have to be moved away from it. If something's good, you can move towards it. But you have to respond to the environment. And you got to be able to transport materials. You have to transport nutrients inside the cell, transport waste outside the cell. So those are some of the characteristics of life. Okay. So reproduction and hereditary. Okay. Produce offspring sexually or asexually. You have growth and development, metabolism, which is chemical and physical like processes. Movement or irritability. You have to respond to an external or an internal stimulus. You have to have cell support. You got to be around the cell to support it. You can't just leak out all over it. There has to be protection of the cell, and you have to have a storage mechanism, like your cell walls, vacuoles, granules, and inclusions. All help store things in the cell. And transport of nutrients and waste. Nutrients in, waste have to go out. Okay. This is a basic structure of a bacterial cell. You can see the cells, all different things are outlined here. This is the cell cytoplasmic membrane. You have the bacterial chromosomes or nucleoid, which is it's not in the nucleus, but it's a nucleoid. It's where all the DNA material is located in the center, but it's not encased in a membrane. You have ribosomes, these little dots throughout the cytoplasm that produce your proteins. Cytoplasm is the inside of the cell that supports all the internal portions of it. Okay. We have what's called an S layer in some bacteria. Okay, it's a monolayer. It's used for proteins, for protection, and it have the bacteria attached to things. Fimbriae are little fibers on the outside, little hair-like fibers. They help adhere to other cells to the surface. You have an outer membrane, okay, which is extra membrane, similar to a cell membrane, but it contains liposaccharides, controls flow of materials inside and out of the cell. Okay. A portion of this are toxic to mammals when it's released. That's usually some of your infections. You have a cell wall with bacteria. You do not have that in human cells, which is a semi-rigid portion around the cell that helps hold everything in and helps protect it. Helps determine the shape of the cell. You have pilus, which is an appendage used for drawing another bacteria close in order to transfer the DNA to it. There's a pilus right here. There's a capsule. Okay, capsule is a coating or layer of molecules sent to the cell, sent to the cell wall. Okay, it protects and allows adhesive and receptor functions. Okay, maybe very tightly or maybe very bound very loosely to the cell. Inclusions or granules. These are these things right here. Okay. Includes they store nutrients. Once they're brought into the cell membrane, they store it in these granules or inclusions. Okay. They store fat, phosphates, okay, which is used to produce energy, ATP, you need phosphate. Glycogen, which is your fat deposits for glucose, they break down for energy. Okay. Deposits in dense crystals or particles that can be tapped into when you need them. That's inside these inclusions or granules. Bacterial microcompartments. Okay, these are protein coated. Right there. They're protein coated packets used to localize enzymes and other proteins in the cytoplasm. And you got plasmids. Plasmids are little rings of DNA. Okay? This is what we use when we do genetic engineering. We introduce, like the insulin gene in the bacteria to produce insulin, or whatever type of the bacteria function that's bacteria to produce. We do that in what's called the plasmid. Okay. It's a small double-stranded DNA molecule containing extra genes. And that's what we use when we do our genetic engineering or manipulations of them. Flagellum is the outer portion here, the tail, has several portions, and it's used to beat or move through the fluid and circular motions, and allows the cell to turn around in circles or flip around or propel itself in a straight line. Provides mobility of the cell, of the bacteria. Okay. Appendages. Okay, we're dealing with bacteria. There's two major types of appendages. There's motility, which is your flagella and axial filaments. Okay, they allow the bacteria to move in a given direction to a different environment. And channels, or pili, which allow the bacteria to connect so they can communicate with one another and exchange information. You have glycolics, which are surface coatings. Those are found in bacterial and human cells. Those are the proteins found inside the cell membrane. The flagella, the fiber that sticks off the cell that gives us motion, okay, has three parts. There's a filament, which is the long part. There's a hook and there's a basal body. The filament is a very long helical structure that's composed of the protein flagellin. Okay, that's most of the little tail there. The hook is the curved sheath that comes off the bacteria, and the basal body is where it attaches to the cell wall. And your filament comes off of that hook right there and extends out. So the filament, the hook, and the basal body. And this is what it looks like. This is where the filament comes out. And this is really long and extends out from here. But this is the hook that supports the filament. And then the basal body connects it and makes sure it's anchored into the cell body. All right, so it allows movement. It rotates 360 degrees. When it spins, it can propel it forward, or if it spins, it can make it go in different directions, kind of rotate it. Okay. There's different types of flagellar arrangements. There's monotrichosis, which is a single flagellum at one end. There's lophotrichosis, which is small bunches emerging from the same site. Okay, so you have one flagella coming out of the end of the bacteria, or you have multiple flagellas coming out that are smaller, all come from the same site. Amphitrochus, where you have flagella at both ends of the cell, and peritrochus, which is dispersed all over the cell surface. Okay, we'll give you different examples of those. This is one flagella where you have the hook here and the basal body, and this is your flagellum that wraps all around. Okay, so one coming out, or you can have two or more coming out from the same spot, or one, or at both ends, a flagellum at both ends of the bacteria, or you can have flagellum all over the place. Just come out the entire bacteria. So those are the four different types and their names. Okay. Responses that the flagella do. Okay, they guide bacteria in a direction in response to external stimulus. So if the environment turns acidic or unhealthy to the bacteria, it propels it out of the environment. Okay. Or it can move towards a chemical symbol or light or something like that also. So like a chemical stimuli is chemotaxis. Okay. Chemotaxis is a gradient with bacteria senses, and it can follow that to a positive center. So it can attract bacteria, or it can be negative, right? Repelling bacteria. Something that bacteria is not nauseous to the bacteria. So chemotaxis is positive or negative. There can also be a light stimulant called phototaxis, where bacteria is attracted to the light, where it moves in that direction. And that's what the flagella does. Okay. It's the signal that sets it in motion, and it spins either clockwise or counterclockwise. If the flagella spins counterclockwise, it's a smooth linear direction. It makes it run in one direction. Okay. If it spins clockwise, then it starts to tumble and spin around itself. Okay, like say it's feeding and there's nutrients around, and then it wants to spin around just to produce the area around to get more nutrients by it. That's what it does. So counterclockwise guides it straight through to a different area. Clockwise makes it spin around the same place, kind of tumbles around. Okay. You can see what tumbling is, the flagella is going to move around, and clockwise rotation, it's going to move in a linear direction. Counterclockwise, I said that backwards, didn't I? Clockwise makes it tumble, so this would be clockwise. And counterclockwise, okay, makes it go in a linear direction. Okay. So this is in response. This is so the bacteria can move linear, it moves around. It can follow a chemical gradient, and it can move in a general direction, or it can just tumble around. This way there's no attraction nor repellent, so it stays in the same place basically. Okay. This one, there's a gradient. You can see it's darker over here, lighter over here. Okay, so this is the chemical gradient. It's going to move towards that chemical gradient. It's not going to shoot just a direct straight line, but it's going to focus, and the flagella is going to propel it in a counterclockwise direction and generally move in this direction. Okay, to follow a stimulus or some kind of phototaxis or chemotaxis. Okay. You can have internal flagella. Okay. Internal flagella are enclosed between the outer sheet and the cell wall, the peptidoglycan. They produce cellular mobility by contracting and imparting twisting and flexing motion. They change the shape of the cell. They can make it squish in one area or expand in another area to increase or decrease its surface area. And there's these little fibers that are in here, and they allow the cell to constrict. When they contract, they constrict the cell and they change the shape of the cell. So which of the following best describes the actions of the prokaryotic flagellum? So this is the flagellum, and your prokaryote means your bacteria without nucleus. It whips back and forth to move the cell. It extends and contracts to move the cell. It rotates to move the cell. It attaches to the environment and pulls the cell. It is capable of all the above, right? We talked about all that. It can do all those things. So that's your prokaryotic flagellum. Okay. Fine proteinaceous hair-like bristles emerging from the cell are called fimbriae. Okay, the difference between fimbriae and flagella, they same thing, except fimbriae are very much smaller. Flagella are huge. These are little fine hair-like particles, okay, that extend out from the cell. So not this thick flagellum, but they're the same basic function. Okay, they allow they come outside the cell and they create motion. Okay, they allow you to adhere to other cells and surfaces. So these bristles come out, they move around, and they'll stick to the surfaces. They're very, very small flagella. And you can see they're nothing near the length of the flagellum that we had. But they're small hair-like structures that allow bacteria to stick together. Pili. Pili are rigid tubular structures made of pilin protein. Okay? They're only found in gram-negative cells. We'll talk about the difference between gram-negative and gram-positive cells in this chapter too. They function as joint bacteria cells for partial DNA transfer called conjugation. Allow DNA to transfer from one bacteria to another. Okay, and these are the pili right here. You can see the small structures that come out and they attach one cell to another cell. And you got all the little fimbriae here on the outside. Okay. But this is your pili that allows you to connect and transfer Okay, pull together the bacteria. Glycolytics, okay, those are found in your cell membranes, those found in human cells also. They're coating molecules external to the cell wall. Okay? They're made of sugars and or proteins. Okay, there's two types of glycolytic layers on bacteria. They can be either a slime layer, they kind of just ooze over the cell, just like the name implies, or they can be in a capsule, which is a more well-defined coating. So the slime, if it's a slime layer, we're going to call it glycocalyx. Okay, they're loosely organized and attached, or a capsule which is highly organized and tightly attached. Okay, this is a slime layer. You can see why it's called a slime layer. You can see it's loosely attached, flows over the cell. This is a capsule, very well defined, much thicker layer, much more defined layer. And the functions of this glycocalyx. Okay. Protects the cell from dehydration, keeps water inside the cells. Okay. So it protects it from dehydration and from nutrient loss. It inhibits killing by the white blood cells, which is our immune response. So it helps protect bacteria from the white blood cell response by phagocytosis. Remember, white blood cells react, they're triggered, they basically undergo phagocytosis and eat the pathogens. This glycocalyx layer, if it's the slime layer or the capsule layer, protect it from this white blood cell response. Okay? And it forms like a biofilm over the bacteria. So these are colonies with capsules. You can see how well defined they are. These colonies are well defined here, or these are colonies without capsules, how they spread out and they're not sticking together as well. Okay. Again, here's the capsule. This is what it looks like under a light microscope. And these are your cell bodies. And the capsule surrounds the cell bodies. All right, the cell envelope. This is an external covering outside the cytoplasm. It's composed of two basic layers. You have a cell wall and a cell membrane. Okay? It maintains the cell's integrity, holds the cell together. Okay? There's two different groups, the different gram stains we talked about, either gram-positive bacteria or gram-negative bacteria. And different antibiotics affect each one of those differently. So it depends on your patient is gram-positive, gram-negative. Depends on which type of antibiotic, which choice of attack you're going to use to kill the bacteria. So gram-positive bacteria have a thick cell wall composed primarily of peptidoglycan and a cell membrane. Gram-negative bacteria have an outer cell membrane, but a thin peptidoglycan layer and cell membrane, so a thin cell wall. Okay. So the cell wall that binds bacteria determines the cell shape, prevents lysis due to change in osmotic pressure. Remember, osmotic pressure is different concentration due to electrolytes or solutes in the material. Okay, like our red blood cells. If we don't mix the right Ringer solution, we can lyse the red blood cells because red blood cells don't have a cell wall, they have a cell membrane. So if you have a membrane here, say with osmotic pressure, okay, say we have a lot of solute molecules inside. Now the solutes cannot travel, only water can travel across that cell membrane. So it's going to try to equalize it. So it's more concentrated inside, so water is going to flow in to equalize the pressure. And it's going to explode. It's going to get larger and larger until it blows up. That's lysis. If the cell has just few and there's a whole lot outside, then the water is going to flow out of the cell membrane and it's going to shrink and become non-functional. When we have a red blood cell and we have the same amount inside as outside, water flows out, water flows in at the same rate. Okay? That's osmotic pressure. What the cell wall does is prevent the bacteria from either blowing up or shrinking to where it's not to die, okay, in environments where the solute is not controlled, like high salt concentrations. And we got bacteria that live in tremendously high salt concentrations in the ocean. Okay? And they do this is the cell wall. This is what protects them. Okay? This is one of the things the cell wall does is prevents lysis or shrinking due to this osmotic pressure from the solute concentrations in your environment. Okay. And peptidoglycan is a major component of your cell wall. It's a macromolecule, repeating framework of long glycan chains cross-linked by short peptide fragments. So it provides a scaffolding or structure that things go onto and protects the bacteria. We good so far?
Peptoglycan protects the bacteria pretty much.
Yeah, it's the main component of the cell wall. Protects it in this way, in the way it prevents bacteria from having problems in different from osmotic pressure, different environments or different solute concentrations.
Okay, thank you.
No problem. Okay. So the structure of the cell wall, since the peptoglycan of the cell wall is a huge three-dimensional lattice work, like a scaffolding. Okay? It's actually one giant molecule and it surrounds and supports the cell. Okay? This shows a molecular pattern of the peptoglycan. It has alternating glycans, NAG and NAM. NAG is N-acetylglucosamine and NAM is N-acetylmuramic acid. Okay? Those are the two alternating glycans that make up this peptoglycan. Okay? It's bound together in long strands. The muramic acid molecules on parallel chains are bound by cross links of the peptide. I'm going to show a picture in just a second here. In large view, the links of the NAM molecules, tetrapeptide chains branch off the muramic acid, connect by amino acid bridges. These amino acids and inner bridges can vary and may be lacking entirely, depending on how the linkage is. It's this linkage that provides rigid yet but flexible support to the cell, because the cell can still change shape. So you still have some flexibility in that membrane. It can't be this thick membrane that is inflexible. It has to give with the cell so the cell can move and change its shape also. Okay? And you can see the different linkages of the different NAM molecules and NAM and NAG molecules here, and how they have crosslinks. And that forms a thick cell wall, but it also maintains flexibility so the cell can change shape. And this is their interbridge with amino acids that connect them together. Okay. So the gram-positive cells have a thick cell membrane. So a thick peptoglycan layer. It's usually 20 to 80 nanometers thick. It includes teichoic acid and lipoteichic acid. Okay. Functions in cell wall maintenance, enlargement, functions during cell division, because you have to be able to constrict it, moves cations across the cell envelope, and stimulates specific immune response. Okay. So the body, as a specific response, when it recognizes a gram-positive bacteria. Some of the cells, some of these gram-positive bacteria have a periplasmic space is between the cell membrane and the cell wall. And that's where these different fimbriae occur. And they allow the cell to constrict and change shape. And they occur in this periplasmic space between your cell membrane and your cell wall. Okay. So this is your gram-positive bacteria. You can see this thick peptoglycan layer. Okay, see how thick that is. You got the teichoic acid. This is your envelope, the entire thing. You have proteins in the membrane. This is your cell membrane below it. Remember, membranes are a it's a diphosphoric dilipid. I'm going to blank with the name, but it's a membrane composed of hydrophobic and hydrophilic ends, and it gives you two layers: hydrophilic on the outside, hydrophobic on the inside. Okay. So this is your cell membrane here. Periplasmic space would be this space right here between the cell membrane and the cell wall. Diphospholipid layer is your cell membrane. That's what it is. Gram-negative. Gram-negatives do not have a thick periglycan layer. Okay? They don't have a thick cell wall. Inner and outer membranes, and 10% between them contains a very thin peptoglycan layer. Okay? The outer membrane contains lipopolysaccharides. The lipid portion is an endotoxin. Okay? Endotoxins can be very toxic when they're released during an infection to the human body. Okay? So these gram-negative cell membranes can release this endotoxin. That's the lipid portion of it. They function as receptors and block our immune response to it. Okay. that contains porine proteins in the upper layer. These regulate molecules entering and leaving the cell. What goes in, what goes out is controlled by these porine molecules in the protein. Those are proteins in the upper layer. Okay. It's popping up. All right, so here's a picture of it. Again, see how thin this peptidoglycan layer is as composed of a thick gram-positive layer. You have porine proteins that allow transforming of things inside out of the cell. This is your cell membrane right here. This is a protein in the cell membrane. This is your cell wall. Okay. Bilipid phospholipid is your cell membrane. Remember, hydrophilic on the outside, hydrophobic on the inside when they're arranged. You still have a periplasmic space, which is between the cell wall and the cell membrane. Okay. So this is a comparison. See the huge peptidoglycan layer and the small peptidoglycan layer. So gram-positive bacteria have a much thicker cell wall. We both have still the same cell membrane. Gram-negative is a thinner cell wall. Okay. Gram-negative bacteria transports a lot of things in and out, can create endotoxins. Okay. Gram-positive, it's much thicker and harder to get through the cell wall. Okay. This is the characteristics of gram-positive, gram-negative. One layer basically, and then the gram-negative is two layers. Okay, the peptidoglycans produce a cell wall very thick. The different acids and polysaccharides make up the chemicals. Lipopolysaccharides, lipoproteins, peptidoglycan, and porine proteins allow transfer of things across that cell wall much easier than the gram-positive. So it's thicker, and this is thinner. There's no outer membrane. There is an outer membrane on the gram-negative bacteria. Periplasmic space is very narrow in the gram-positive because it's such a large layer, it squishes it down, and it's extensive in the gram-negative. Okay. Gram-positive is permeability to molecules is more penetrable and then less penetrable. Which of the following is not found in gram-negative cells, ball structure? Which is only found in gram-positive? Okay. That would be your teichoic acid, teichoic acid. Everything else is found in both gram-negative and gram-positives. Okay. So teichoic, this one is only found in gram-positive. The gram stain, this is how we tell whether it's going to be gram-positive or gram-negative. They stain differently. So different stains that distinguishes cells with a gram-positive cell wall from those with a gram-negative cell wall. So gram-positive, they retain crystal violets and stain purple if it's gram-positive. Okay, gram-negative lose the crystal violet and stain red from the saffron counterstain. So gram-positives will stain purple color and gram-negatives will stain a red color. So you can tell which cell or bacteria you're dealing with. Okay. This allows us to classify the different bacteria and identify them, which we have to do to be able to treat somebody. We got to know what we're treating before we treat. So it's an aid in diagnosing infection and guiding drug treatment, whether it's gram-positive or gram-negative, because they're going to react differently because they're different structures to different antibiotics. Okay. Then you get some non-typical cell walls. They're not gram-positive, gram-negative, they're non-typical. Some bacteria lack typical cell wall structure. So mycobacterium and nocardia. Okay. Gram-positive cell wall structures with lipid mycolic acid, which is a cord factor. Pathogenicity and high degree of resistance to certain chemicals and dyes. And it's a basis for acid-fast staining, used for diagnosing infections caused by these types of microorganisms. Okay. Some have no cell walls at all. Mycoplasma has no cell walls. So you can't use an antibiotic like penicillin. Penicillin puts holes in the cell's wall, puts holes in cell walls and kills bacteria that way. So that won't work with a mycoplasma. Cell wall is stabilized by sterols and it's pleomorphic design. The phospholipid bilayer, this is your cell membrane. Remember, you have your phosphate groups, which have a charge, and then your long chain fatty acids inside. And we just line these up. Okay. So this is hydrophilic on the outside, where it comes in contact with the environment and inside the cell. And an internal layer here is hydrophobic. It's all your fatty acid chains that don't bond to water. Okay, so it functions, the cell membrane provides a site for energy reactions, nutrient processing, and synthesis. It allows the passage of nutrients into the cell, allows waste products to leave the cell. And it's selectively permeable, which means only certain things get in and only certain things go out. It's selectively permeable. This is your cell membrane. This is a better picture than I can draw. But these are all your phosphate heads here, and these are your fatty acid chains. So it's all negative charges here, and there's neutral charges inside. Okay. You got glycolipids, you got transport proteins, which allows you to grab things and push it inside something like glucose. Okay. Different gateways and things like that allow waste to go out of the cell. All right. The cytoplasm. Cytoplasm is what everything's suspended inside the cell. It's a gelatinous solution. Sugars, amino acids, salts. 70 to 80% of it is water, but it's a little bit thicker than water. Okay. Serves as a solvent for materials to use in all cell functions. It's where all the reactions occur in the media, that all reactions occur inside the cell. Okay. The nucleoid. Now, bacteria do not have a nucleus, but they have a nucleoid. Nucleoid is just in the center of the cell where the DNA material is. It does not have a double membrane around it. That's what separates it. So the nucleoid is the area in the center where the DNA is inside the bacteria. The nucleus is where it is in a human cell or animal cell or plant cell, where the actual nucleus is a double membrane. This has no membrane around it. But it's kind of in the center where all the DNA accumulate. Okay. So you still have chromosomes, which is DNA. Single circular double-stranded DNA molecule is your chromosome. Contains genetic information that's required by the cell to perform all its functions, make its proteins, make its enzymes, and exist. And it also allows it to split and transfer that information into the daughter cell so the next bacteria has the same DNA information. Now, plasmids, we said, were those small circular rings inside the cytoplasm. This is what we use when we do genetic engineering. We might put like an insulin gene inside there or some other gene. But they are freestanding and not tied into the chromosome. There's free, small, circular, double-stranded DNA. They're not essential to bacterial growth and metabolism. They're used in genetic engineering. That's where we attack everything into. We can manipulate them very easily, and they transfer from cell to cell. Okay. So we can transfer from one cell to another this plasmid after we've made its chains. Okay, these are nucleoids. Again, this is your DNA kind of in the center of the cell. There's no nucleus around it. Okay. But it's just the material inside the cell. That's your nucleoid. The ribosome. Ribosome is where proteins are made. It is where the information from the DNA goes into messenger RNA, okay, travels out to these ribosomes. The ribosomes convert DNA information into proteins. There's two subunits to every ribosome, a large and a small. And think of it like a stapler. Big unit, small unit, clamps down and adds an amino acid. Clamps down again, adds another amino acid. And this amino acid chain is our protein. It's just like staples one to each one between these large and small subunits. Prokaryotic is different from eukaryotic ribosomes in size and number of proteins. It is the site of protein synthesis, just like it is in all animal cells also, well, eukaryotes. It is found in all cells. All cells have to make proteins. They have to do something to make their cell walls, to make the function, to make enzymes, everything that they need for their organisms, for the organisms to function, whether it's multicellular or unicellular. But this is a ribosome. This is a complete ribosome. This is the big unit. This is a small unit. This thing opens up right here, and a little transfer RNA comes in here with an amino acid attached, and it staples it, staples it, staples it, and makes a polypeptide chain. Okay, so think of ribosomes as big staplers that staple amino acids into a chain. big unit and the two subunits, large unit and small subunit. All right, inclusions and granules. Maybe we store things inside. Those are inclusions and granules. There's interstellar storage bodies. They vary in size, number, and content. Okay? Bacteria cells can use them when environmental sources are depleted. Okay? They can store stuff inside these inclusions for use later on when they need nutrients for energy. Okay? These are you can see the inclusions right here in your bacteria. And they can all line up. They can be in different areas of the cell, depending on how the energy needs of the bacteria. And they also have what's called a cytoskeleton. This is the scaffolding inside the cell that maintains structure. Many bacteria have an internal network of protein polymers that's the closest to the cell wall. They help with moving materials to the cell wall. So when things have to come across that cell wall, so this is your cell wall here, and then you have to have a scaffolding, these little proteins here, and these run into your ribosomes and your Golgi apparatus and things like that. And material can transport like waste products and then go export across the cell wall. So this allows like a freeway system. The little scaffolding is an internal network, protein polymers inside the cell, and it's associated with the cell wall. That's what they do. You can see them lighting up the fluoroscopy. You can see the structure of the cytoskeleton there. And they're made of actin filaments. Remember, actin and myosin cause constriction of your muscles. So now they allow the cell to pull into different areas, different shapes. Endospores we talked about. Endospores are when the bacteria goes into I don't want to say hibernation, but it stops basically living. Then it can come back through a vegetation phase. Endospores are the most difficult things to kill. Okay. Sterilizers in medical and dental offices, we use little packages with endospores in them. We put it in there, we run our load. Once a week, we send it to the government or to the CDC. They analyze it and make sure the endospores are killed. Because if we kill the endospores, we know we're killing everything else on our instruments. And we're getting rid of all the bacteria. If we're getting rid of endospores, we're getting rid of everything because those are the toughest to kill. They're inert, they're resting cells, just like hibernation. They're produced by some gram-positive genre bacteria, Clostridium, Bacillus, and Sporosarcina. They have two-phase life cycles. The vegetative cell, which is metabolically active and growing. That one's a regular bacteria. Okay. The endospore. The endospore is when exposed to adverse environmental conditions, they shut down. They stop reproducing, they go into hibernation. They're very high resistance and very long-term survival. Okay. Most difficult to kill. Okay. Formation of the endospore, they're the hardiest of all life forms. They withstand extreme heat, drying, freezing, radiation, chemicals. They don't die. Okay. But we can kill them. But they are not a means of reproduction. When they're the endospore, they cannot reproduce, they cannot feed, they're just existing. Okay. They're shutting down until the environment is better for them to come up, go back into a vegetative form. So when they germination, they return to the vegetative growth and start multiplying again and living. Okay, and this is a picture of the cycle. This is a vegetative cycle where it comes, and this is how it condenses itself into an endospore. And this is the endospore where everything's completely compact. Okay, the free spore is released when the dead spermicide falls away. So it's continuing to perpetuate bacteria even though it's dead. And then it goes into a vegetative state when it gets in favorable conditions again. Okay. So they're dehydrated. So you can't dehydrate, you can't dry them out. They're metabolically inactive. They're not producing anything. They're very thick coat. Longevity is basically immortality. They live maybe 250 million years like that. That's pretty much immortal. If you're gonna live 250 million years, I would classify that as immortality. They're resistant to cleaning, boiling. The only way to kill them is with a sterilizer. Pressurized steam, 120 degrees centigrade for a minimum of 30 minutes will destroy them. And that's what our sterilizers in medical and dental offices run it. They pressurize steam, 120 degrees centigrade, and they leave it that way for 30 minutes. And then we've killed everything. And you check and make sure they're still doing your units. Okay? If you discover the bacteria you're studying store is phosphate, which of the following would you expect it to contain which of the following would you expect to contain the stored phosphate granules? Okay. Where are phosphates stored? And it's already answered for you. I didn't see that. I'm gonna go back and fix that. I mean, ribosomes do what? They make proteins, linking amino acids together. Plasmids are your ring structure of DNA that allows you to do genetic engineering. That's transferred from cell to cell. Okay. Liposaccharides are part of your cell membrane. Endospores we just talked about. Granules are the things that incorporate into those vacuoles, and they're stored. Phosphates are stored there. Fats are stored there. Glycogen is stored there for energy uses. Okay. Phosphates allow you to create ATP. It allows you to go from ADP to ATP with phosphates. Okay. So those are stored in these as granules. So we can use them for energy. Okay. Bacterial shapes, arrangements, and sizes. Bacteria comes in all shapes and sizes, and all kinds of names. I want you to be familiar with the names so you can look them up on the exam. You can look it up in your book. You don't have to memorize everything. I don't have all these memorized. And hundreds and hundreds or thousands and thousands of these bacteria. But I want you to be familiar with them enough to look it up and identify it when you see it. So you can look up a textbook and understand what we're talking about. So they vary in shape, size, and arrangement, typically described by 1 to 3 basic shapes. If they're coccus are spherical, they're ball-shaped. Bacillus are rod shapes. Okay. Coccobacillus are short rods. Okay. Vibrio bacillus are gently curved. Spirillum, helical, a comma or twisted rod. Okay. Spirochetes are like a spring. Those are spirochetes. They're spirillum. Okay. Those are the basic shapes. Coccus are round, bacillus are rods, and spirillum are spirals. This is coccus. These are round shapes. If you see this on a nursing exam, you would choose the bacteria that has the word coccus in it. It'd be a long name, but you know coccus means round or spherical. Rod shaped are bacillus, and you see the different shapes the rods take on.
Didn't you say, isn't tuberculosis rod shape?
Yes, I believe so. Tuberculosis is a bacillus. I'll double check. I believe tuberculosis is rod shaped also. Vibrio are curved. See how they curve around there? You can see the curves in the bacteria. Spirillum are spirals. You can see how the spirals go across here. And the spirochetes are like a spring that wind. They go around. And you also have branching filaments, which are, you can see just like we had with glycogen storage. And then they branch. And the cocci build up on that. So you have all different types of shapes. Those are just some basic ones. Okay, so the first one we looked at, it was Staphylococcus aureus. The second one was Legionella pneumophilia. Fibrocolor was the third one. Aquaspirillum was the fourth. Borrelia burgdorferi was the fifth. And Streptomyces species was the sixth one. I'm going to just take a look at it. So variation in cell shape and size within a single species. Some species are known for pleomorphism. Pleomorphism means they can take on different shapes in the same species of bacteria. Okay. So you can have different arrangements. You can have metochromatic palisades where they all line up together. Okay. Or pleomorphism, where they can have different shapes all together. All the same bacteria. You're not limited to just one shape. The arrangement of the cells depend on the pattern of division and how cells remain attached after they divide. Cocci one, remember cocci means spherical. They can be single. They can be diplococci, which means two, a pair. Tetrads, which are groups of 4. Irregular clusters, chains, or cubical packets. And all the different ways cocci can manifest themselves as different shapes. The bacilli are rod shape, but they can be diplobacilli, which is two combined. They can form chains or palisades where they chain off from a single point and group together. Okay? Or chains that can link together. Okay? They all take on different shapes. Okay? This is diplococci, which is two cells joined together. This is streptococci, which is a chain of spheres. Okay. Tetracocci are four spheres in a group, or you get packets of 8 to 64, like a crystal shape. Okay. You can be in several different planes. You get an irregular cluster in different planes, a staphylococci or a micrococci. Okay. Bacterium are very small, what we're dealing with. This is a human hair. This is the size of the next step. This is a pollen. This is a red blood cell. This is a lymphocyte. And down here are your bacteria. Okay, these are Staphylococcus down here. And this is E. coli. So you can see how small bacteria are in comparison to the rest of things in the human body. And viruses are even smaller than that. This is a rhinovirus right down here. Even smaller than bacteria. Okay, we can classify these different prokaryotes. We can classify with microscopy, macroscopic morphology, which is how they look to the naked eye, the physiology, how the bacteria react, serological, how they in different environments, like in blood, and genetic and molecular analysis with DNA. So we got different ways of classification of these prokaryotes. And we have a manual called Bergey's Manual of Determinative Bacteriology. It's a 5-volume resource covering all known prokaryotes. Huge manual, 5 big volumes. Do not memorize that whole manual. Classification is based on genetic information. It's called phylogenetic. Okay? There's 2 domains. There's archaea and bacteria. Those are your prokaryotes. There's 5 major subgroups within 25 different phyla. Okay, this is some of the examples, some examples from the book. Okay, volume 1 is a domain archaea. Includes your prokaryotes with unusual morphology, unusual shapes, ecology, and modes of nutrition. Okay. Many of the known members are adapted to a habitat with extreme temperatures, hypothermophiles, or salts, like we talked about before, halophiles, okay. Acidities, acidophiles, pressure, lack of oxygen, anaerobic, and sometimes a combination of these. So these are ones with unusual shapes that live in very extreme conditions. Okay. Great ecology important to its actions in biochemical cycling. Okay. And we'll talk about more in section 4.7 kind of in the book there. Okay, phylum Crenocreota. Members depend on sulfur for growth. Okay, not oxygen. Okay. May inhabit hot and acidic sulfur pools and vents. Some species have adapted to cold habitats. Okay. Phylum Nano, Archaeota. It's a very newly discovered group, extremely small archaea found in the salt mines and caves. They're all over the place. Phylum Urarchaeota, the largest and best studied group of archaea. Okay. They include methane production, they produce methane gas, methanogens, high saline or salt, halobacteria, or high temperature thermophiles. And they work by the reduction of sulfur compounds, not oxygen. Okay. So they live in different environments. Okay. This is a thermophilic, so I say from the deep sea thermal vents. And these bacteria that live under very extreme conditions. You can see it has different shapes to it. Some are rod shaped, some are short rods, some are spherical. But 160 degrees Fahrenheit is pretty warm for bacteria to live. Okay. Methanocaldococcus is a hyperthermophile inhabiting hydrothermal vents at near boiling temperature, near 100 degrees centigrade. And you can see how they kind of morph and take on different shapes as their own. I mean, this looks like a cocci here, but then you see how it's morphed into two of those. How it takes on a ragged shape there. So it can exist at very high temperatures. 230 degrees Fahrenheit or 100 centigrade. All right, in volume 1, domain bacteria 1. This domain contains deeply branched and photosynthetic bacteria. Okay. Consider the oldest evolutionary life still in existence. Contains a wide diversity of different common bacteria. It's called photosynthesizers. They use the sunlight for energy, which is our cyanobacteria like algae, green blue algae. Thermophiles, radiation resistant bacteria, halophiles for salt and sulfur metabolizers. There's a phylum Aquaficae, which contains small thermophilic rods that inhabit underwater volcanoes. They exist in all kinds of environmental conditions. Okay. Continuing with the book, you have phylum Thermotogae, okay, which is similar to the one we just talked about. It includes thermophilic halophytes, halophiles, which are salt and high temperatures. They live in deep sea vents, very high concentrations, very high temperatures. Okay. Phylum Chlorobi, known as green sulfur bacteria. Okay. This phylum consists of anaerobic bacteria that live without oxygen, live in the muddy layers of lakes and ponds, where they photosynthesize and metabolize sulfites. Okay. They live without oxygen, they metabolize sulfur instead of oxygen. All right, let's continue with the table. That we have phylum Deinococcus, as a thermos, which is a small phylum of extremophiles. Okay, extreme temperatures or pressures, ranging from being highly radiation and desiccation resistant, live in the soil, fresh waters, and in very hot aquatic habitat, like hot springs and pools. Okay, phylum Cyanobacteria. Again, they're photosynthetic. Okay, found in aquatic habitats on the soil, associated with plants, fungi, other organisms. Blue, green, yellow, red, and orange in color. And this is what some look like. These are tiny anaerobic halophilic rods. You see some of the rod shapes, but they're halophilic. That means they can withstand high salt concentrations and they reduce sulfur compounds. Okay. Trichia is a cyanobacteria. You can see the green and yellow colors of it. Okay. Forms these colonies, these radiating fibers that float in your lakes and ponds. Okay. Volume 2 for this book contains domain bacteria 2. This domain, along with the phyla, are covered in volumes 3, 4, and 5, contain bacteria that have the greatest medical impact for humans. Okay. Phylum Proteobacteria contains 5 classes, representing extremely great cross-section of over 2,000 identified species of bacteria. Okay, they all have characteristics of gram-negative cell walls. Remember the thin cell walls contain bacteria with a wide range of adaptations, shapes, habitats, and ecology. Medically significant members include the one that cause rickets, okay? The parasitic rickettsiosis, okay? E. coli, Neisseria, okay? Salmonella, the vibrio species of it. Bacteria live in the intestinal tract of animals. Okay? Myxobacteria have unique characteristics of being the only bacteria that can aggregate into multicellular structures. So bacteria can form multicellular structures. This Myxobacteria. This is the one that causes Rocky Mountain spotted fever. This is rickettsia cell. Okay. You can see this is rickettsia cell right here. And this is a phagocytic white blood cell that's eating it to dissolve that cell, dissolve that bacteria. Okay, this is E. coli. It's a foodborne pathogen, causing contaminated food, causes severe illness found in humans. Okay, this is a thermophile, desulfovibrio. Okay, this is living in a pond. This is biofilm, a pond. Your vibrio shapes takes on little bends and angled shapes of your rods. Firmicutes, okay, mostly gram-positive bacteria. This one's gram-positive, the other one's gram-negative. Okay. A low G plus C content, guanine to cyanine ratio. So you do a DNA analysis, you see remember guanine to cyanine, adenine bonds to guanine, adenine bonds to thymine, cytosine bonds to guanine. So if you compare this ratio, you can tell which bacteria have similar DNA makeup, okay, by analyzing that ratio. Okay. These are endospore forming, generally include bacillus and Clostridium. Remember, endospore formers are toxins, very hard to get rid of. Okay, found this genera are your Staphylococcus and your Streptococcus. And you can see Anthracus, Bacillus anthracus. Okay, this is scan electron microscopes from the rod shape next to a red blood cell. Here's your blood cell, and this is cells that cause anthrax, bacteria that cause anthrax. Okay, Streptococcus pneumoniae, display the Diplococcus, see the two together. So it's a Diplococcus remnants of the species of bacteria. In volume 4, in the phylum Actinobacteria, this is a category which includes a high guanine to cytosine ratio. Okay, that's how they're related by DNA analysis. These are also gram-positive bacteria. They differ in their morphology and life cycles. Prominent members include branching filaments, actinomyces, spore-bearing Streptomycetes, Corninium bacterium, okay, Mycobacterium, and Micrococcus bacteria. Okay. This is Streptomyces, common soil bacteria, often the source of antibiotics that we can create. You can see the different cocci forming chains as they come across. Mycobacterium tuberculosis. That was a tuberculosis question. It's a bacillus. Bacillus means it's rod shaped, and this is the bacteria that causes tuberculosis. Okay. So you were right with your question on the bacillus being rod shaped for tuberculosis. Mycobacterium tuberculosis. Okay, volume S represents a mixed assembly of 9 phyla. Okay, they're all gram negative, that they widely vary. Okay. Phylum Chlamydia, okay, a group of intercellular parasites that reproduce inside host cells. In other words, chlamydia cannot reproduce on its own; it has to be inside of host cells. It's a parasite. These are among the smallest of bacteria with a unique mode of reproduction. Several species cause disease of the eyes, reproductive tract, and your lungs. Example of this is chlamydia, one disease it can cause.
Isn't that like a STD?
Yes, it definitely is an STD. That's how it's transmitted. And we'll show some examples of that too as you go on too. That's a perfect question. Phylum Spirochetes, bacteria distinguished by their shape, okay, and mode of locomotion, how they move. They move, their movement twisted, or slender twisted cells by means of a periplasmic flagella that comes out. They live in different habitats, including the bodies of aminals and protozoans, fresh and marine water, and even muddy swamps. Planctonomycetes live in fresh and marine water habitats, reproduced by budding, like breaking a piece off of their of the bacteria. Many have a stalk they use to attach to a substrate to hold on. Unique features having a membrane around their DNA and special components that close in that membrane. Okay. So they do have a membrane around their DNA in these Planctomycetes. We've let it speculate that they're similar to ancestral form that gave rise to eukaryotes, because eukaryotes have that double membrane from the nucleus. So it's one of the evolutionary steps from bacteria to eukaryote, from prokaryotes to eukaryotes. So prokaryotes were long before eukaryotes. Bacteroidetes, okay, widely distributed. These again, gram-negative anaerobic rods. Anaerobic means without oxygen. Okay, inhabiting soil, sediments, water habitat. Frequently found as normal residents of the intestinal tract of mammals. They're grouped by the rate of Fibrobacteries and Chlorobia. Several play a major functional role in the human gut. Some are involved in oral and intestinal infections. Example is Bacteroides. Okay. This shows you an infected cell revealing the vacuoles. Remember, the vacuoles inside the bacteria is where they store the materials when they're ingesting them. This is a chlamydia cell in various stages of development. Again, that's an STD transmitted disease. This is M. Treptonema pallidum. These are spirochetes that cause syphilis, another STD, sexually transmitted disease. Okay, that's what syphilis bacteria looks like. Gemata is a budding cell through a microscope, and you can see the blue nucleus here. The nucleus is the center where the genetic material is. You see the green surrounding area where it's budding, making that as cocci chain. Bacteroides species may cause intestinal infections. These are all bacteroids. You see the rod shapes, and different species of these cause intestinal infections. Some are tuberculosis is one of those that cause a respiratory infection. The G plus C base composition, again, guanine, cytosine composition, determine what's related with the DNA. The overall percentage is an indicator of relatedness, which bacteria is related by their DNA, by their GC ratio. Okay. Bacteria with a significant difference in GC ratio percentage are less likely to be genetically related. Ones that have a close GC ratio are more likely to be related. It's another way of classifying our bacteria. Okay. So the domain archaea, we have archaea and prokaryotes. Archaea are primitive, adapt to extreme habitats and modes of nutrition. The domain is under bacteria. The phylum Proteobacteria are gram-negative cell walls. The phylum Firmicutes are mainly gram-positive with a low GC ratio, and phylum Actinobacteria are gram-positive with a high GC content. So those are the different types or the taxonomic groups where the bacteria belong, these archaea. And this is a chart that shows the different bacteria, archaea, and these are your eukaryotes. And you can see you have some overlap here where you get some of that membrane being formed in the archaea. These are all your bacteria here. And these are also considered bacteria, and then into your eukaryotes. Because all these ones, all the ones over here, these are all prokaryotes. Diagnostic schemes for medical use. We use the phenotypic qualities and identification part. We have to identify the bacteria and be able to know what it does to treat our patients correctly. Restricted to bacterial disease agents. Devise bacteria based on cell wall structure, their shape, their arrangement, and their physiological traits. So we know how to treat patients with these infections. Important families in general of pathogenic bacteria and different nodes of some diseases. So bacteria with gram-positive cell walls. Okay, gram-positive cell walls. Okay. And cocci and clusters or packets that are aerobic, which means they use oxygen. Okay. Family, Staphylococcus, Staphylococcus, members of cause boils and skin infections. That's your Staphylococcus. And you can see groups of 4, groups of 8 of the different cocci. Okay. Cocci in pairs. Streptococcus causes strep throat and dental caries. And again, Diplococcus or in chains. And aerobic cocci.
What are dental caries?
Cavities
Oh,
in the teeth. Dental caries are cavities in the teeth. They eat. What they do is they feed on the sugar, the sucrose in the body, and they have a very thick biofilm, and they stick to the teeth. And what the bacteria do is when they feed on sucrose in the mouth, they produce acid. This acid destroys the enamel and kind of cuts through the tooth. So you have a tooth here. There's your tooth. So you get this film, and then you got dent inside the tooth. I didn't mean to do that one. You got the dent layer inside, and then the nerve inside of that. Okay, so this is a biofilm that adheres here, and once you get sugar, sucrose, it starts producing acid. So the byproduct of acid eats the enamel, so it starts to destroy the enamel, and it starts to feed in through here. And you get this little, and that's a dental cavity. So it's up on an X-ray. Okay, so this is where the Streptococcus, the bacteria is not dissolving the enamel. The acid produced by the Streptococcus is what destroys enamel and eats into the tooth. Depending how far it goes, depending how much pain you get and what treatment, whether it needs a filling, crown, root canal, whatever it needs. That's what dental caries are cavities in the teeth. Okay. Anaerobic oxide without oxygen. Okay. These are peptococcus. Okay. They're involved in deep wound infections. When you get gangrene, stuff like that, you get really bad infections because there's no oxygen in those areas. Okay. Spore-forming rods. Okay. Bacillus causes anthrax. Clostridium causes tetanus, gas gangrene, and botulism. All nasty stuff. Those form spores. Non-spore forming rods. Okay. Lactobacillus. Listeria is food infection. This is Propionibacterium. This causes acne. Corneae bacterium causes diphtheria. Mycobacterium causes tuberculosis and leprosy. Those are different rod shapes there. Okay. Nocardia. Nocardia causes lung abscesses in your patient. Okay. Actinomyces. Actinomyces cause dental infections in the mouth. Streptomyces. Source of antibiotics. Use that to create antibiotics to fight different bacteria. Okay. Bacteria with a gram-negative cell wall. Okay. We'll use on these now. Then we have Neisseria, which causes gonorrhea and meningitis. Okay. Those are those diplococci. So both two cocci involved in that. Okay. Moraxella, Actinobacter. Again, those are aerobic. Anaerobic cocci. Villanella, which causes dental disease. It's more dental caries. Pseudomonas causes pneumonia and causes infections in burn patients. Miscellaneous rods. Brucella causes undulant fever. Bordetella causes your whooping cough. Francisella causes tularemia. Coxiella causes Q fever. Legionella causes Legionnaire's disease. So it's important to isolate the bacteria causing infection because then you know how to treat it. To isolate, you have to identify it. Facultative rods and vibrios. Okay. We have different types. You got Salmonella, which causes typhoid fever. Shigella, which causes dysentery. Okay, other different ones. Yersinia, which is one that causes plague. Vibrio is cholera and food infection. Campylobacter causes enteritis, infections in the intestines. Helicobacter causes ulcers in the intestines. Pasteurella causes bite infections. All different types of genre there. Okay. Bacteroides. Fusobacterium causes anaerobic wound and dental infections. And anaerobic wounds are without oxygen and they fester. These are helical and curiform bacteria. Treponema causes syphilis. Borrelia causes Lyme disease. Leptospira causes kidney infections. And those are helical form bacteria. Intracellular bacteria. Rickettsia causes Rocky Mountain spotted fever. Ehrlichia causes human ehrlichiosis. Chlamydia, like we said, was our sexually transmitted disease, one of them. Bartonella causes trench fever and cat scratch disease. Bacteria that do not have cell walls, mollicutes. Mycoplasma causes pneumonia. Ureaplasma causes urinary tract infections. Okay, we have species and then subspecies. So species is a collection of bacterial cells which share an overall similar pattern or trait, in contrast to other bacteria whose patterns differ slightly. That is a species. Then the strain or variety is a culture derived from a single parent that differs in structure and metabolism from other cultures of that species. Those are bivars or morphovars. Those are a strain or variety of the bacteria that have branched off. And a type. A type is a subspecies that can show differences in antigenic makeup, like your serotype or serovar, susceptibility to bacterial viruses like phage viruses, and pathogenicity, different pathotypes, how susceptible it is to different other types of infection. All right, so round bacteria cells growing in irregular clusters will be best described as
Staphylococci.
what's that?
C.
So staphylococci? Yep. Because that shows you the cocci because it's round and it's irregular clusters with staphylo like a rod shapes with little irregular shapes. Very good, perfect. Okay. Prokaryotes that have unusual characteristics. Okay. Free living, non-pathogenic bacteria do not cause infection. Photosynthetic bacteria use photosynthesis, consists of nutrients from inorganic compounds. Okay. Cyanobacteria like blue-green algae, green and purple sulfur bacteria, and gliding fruiting bacteria. Okay. Cyanobacteria, these are gram-negative cell walls, extend to thylakoids, which produce, capture the sunlight, energy, with photosynthetic chlorophyll pigments and gas inclusions. Okay, so it allows you to capture the energy of the sun. These are cyanobacteria. They come in different colors. You see orange ones there. These are found in lakes and streams. These are the cyanobacteria green color. And this is a stromite pattern with different layers, basically. This is what they look like. You see this triasian pattern in them. Green and purple sulfur bacteria are photosynthetic. Okay. They take sunlight and create energy. Contain photosynthetic pigments, bacterial chlorophyll. They do not give off oxygen though. They use the products to produce nutrients for the cell, but they do not give off oxygen as a byproduct. Now, trees and plants in the eukaryotes, they give off oxygen as a byproduct. Okay, they create carbon dioxide and water to turn into glucose and oxygen. That's trees. Green and purple sulfur bacteria live on the surfaces of ponds, and they produce their own energy from the sunlight. Gram-negative, this is a fruiting body of it, and it goes down through the myxospore. Okay, and the myxospore is like an endospore. Okay, and then it starts to go to vegetative growth. It becomes activated. It keeps growing into it and forms an aggregate. This is a colony of these gram-negative cells. Obligate intercellular parasites, which means they live within the cells of their host. Rickettsia is a very thin gram-negative bacteria. Most of them are pathogens, cause disease. And they're intracellular pathogens. They live within the cells. They cannot survive or multiply outside the host cell. So if they're exposed to tabletop, exposed to air, they die. They live inside the host cell. Okay? And they cause Rocky Mountain spotted fever, rickettsia. Rickettsia. Okay. This is the rickettsia cells. Okay, this is the nucleus right here. This big thing's the nucleus of a human cell. Okay, and these are rickettsia that are living inside the cell. And they use the ribosomes and machinery of our cells to trick it into making its own proteins, not decoding our own DNA. It uses its DNA to decode into our ribosomes. Okay. Chlamydias, we talked about. Those are our STDs. Okay, chlamydia is a very tiny bacteria. It's an intracellular parasite. It's not transmitted by arthropods, it's transmitted sexually. Chlamydia trachomatis, so it causes severe eye infections, one of the most common sexually transmitted diseases, and also can cause lung infections. Okay, archaea are the other prokaryotes we talked about a little before that. Those are kind of the crossover points between prokaryotes to eukaryotes. The concept of the third domain is archaea. Okay. Most closely related to eukaryotes than to bacteria, because there is a single membrane around the nucleus, contain unique genetic sequences in their ribosomal RNA, have unique membrane lipids and cell walls. Okay. So you have bacteria, you have archaea, and you have eukarya, which are human cells. This is the crossover point between them, evolutionary. Okay, but they don't have a nucleus, but they have a thin layer of cell membrane around the nucleus. They don't have a double membrane like eukaryotic cells. Chromosomes are similar to prokaryotes, bacteria. Okay, type of ribosome is just like bacteria. This ADS is another one. Unique ribosomal RNA is both in bacteria, archaea, and in eukaryotes. Number of RNA sequences that share with eukaryotes is like share 1. This shares 3 with eukaryotes. Okay. Presence synthesis similar to eukarya. It's positive. It's similar to the eukaryotic cells. Presence of peptidoglycans in the cell wall. Your peptidoglycans are your cell wall, the thickness of the cell wall. Bacteria have a cell wall. Archaea do not, and eukaryotic cells don't have a cell wall. Cell membrane lipids, fatty acids are in fatty acids and ester linkages are in bacteria. Long chain branch hydrocarbons with ether linkages are in archaea. And in eukaryotes, you have fatty acids with ester linkages. Okay. And then there's different exceptions on the sterols in the membrane. Okay, archaea live in most extreme habitats in nature, extremophiles. They're adapted to heat, salt, acid, pH, pressure, atmosphere. They include methane producers, hyperthermophils, extreme halophils, salt, and sulfur reducers. Okay. Archaea live in all different areas. This is all different lakes you can live in. This is archaea under fluoroscopy. Okay, organisms in the domain archaea have peptoglycans in their cell wall. True or false?
False.
False. They do not. The only thing that has peptoglycans is
They're in eukaryote.
prokaryote, right? Because they make the cell wall. There's no cell walls in archaea or eukaryotic cells. No, answer is B, it is false. Peptoglycans are found in cell walls. That makes you, and gram positive, they're really thick. Gram negative, they're really small, they're really thin. Okay, and in the archaea, there are no cell, just like in eukaryotes, no cell wall.